Welcome to the Nonfiction Insights channel.
Thanks for having me.
Yeah, so like I said, we're talking about lifespan, why we age and why we don't have to, by David Sinclair.
Right.
And this book really dives into the cutting edge of longevity research.
It does.
And so, we're going to try to give you a cheat sheet to understanding these concepts.
Yeah.
He really starts the book off in a way that I didn't expect
Oh, yeah.
by talking about his grandmother.
That's right. He starts with a really personal story about his grandmother, Vera.
Yeah, and I like I said, I wasn't expecting that, but what did you think about those opening stories?
Well, I think it's really effective because he grounds this scientific exploration in a personal experience.
Yeah.
It makes it relatable right from the start.
Totally. And what resonated with me was this contrast that he draws.
Oh, yeah. What do you mean?
So, his grandmother, Vera, she lived a long life, but she seemed to lose, her essence
Right.
years before her physical death.
He says that the person she truly was had been dead many years.
And that was the line that really stuck with me because it made me think about, what we even consider to be a long life.
Right. Is it just about the quantity of years?
Or is it about the quality?
Exactly.
Like, are those years healthy? Are those years vibrant? Are those years full of life? Yeah.
And that's what we're going to be exploring in this deep dive.
Totally. And then he goes on to talk about this interesting shift in how children understand death.
Oh, yeah.
Like how around the age of 5 to 7, they start to realize that death is this universal thing.
It's not just something that happens to other people.
That it's going to happen to them too.
Right.
And these reflections, they really set the stage for that core question that he's trying to answer.
But why?
Why do we age?
Yeah, why does this happen at all?
Like, why is this a thing?
Right. And as he points out, most scientists haven't really focused on that fundamental question.
No, they've been too busy.
They've been more focused on treating the diseases that come with age.
Right. He calls it the whack-a-mole approach.
I love that analogy.
It's very true, though, right? Like, we wait until something pops up.
Right. We're not looking at the underlying cause.
And then we just try to, fix that one thing.
Yeah, we're just trying to whack those moles as they pop up.
Exactly.
But we're not addressing the underlying reason why they keep popping up.
And I think that that's what's so compelling about this book.
Yeah, he's really shifting the focus.
He's challenging us to think differently about aging.
To look at it as a process that we might be able to influence rather than just something that happens to us.
And that really ties into this concept of healthspan.
Right.
It's not just about living longer.
It's about living healthier for longer.
Exactly.
Yeah, and it's those healthy years, those active years, that really matter.
So if we're looking at aging as a process, we need to understand the theories that have tried to explain it.
That's right. Let's delve into some of the history here.
Okay, so one of the earliest theories was this idea that aging is just a result of our genes accumulating mutations over time.
Right, kind of like wear and tear on a machine.
Yeah, like our DNA is getting copied over and over again, and mistakes happen.
And those mistakes lead to the decline we see with age.
But then we had things like the cloning of Dolly the sheep.
Yeah. What did Dolly's existence tell us about that earlier theory?
Well, it kind of threw a wrench in the whole thing.
How so?
Because if aging was just about our genes getting messed up over time, then a clone made from an older animal should have been born old.
Right, it should have inherited all those accumulated mutations.
And should have aged prematurely.
Right.
But Dolly didn't really do that.
No, she lived a pretty normal lifespan for a sheep.
Which suggests that there's more to aging than just this simple accumulation of genetic errors.
Yeah, it challenged that idea that our original genetic blueprint is the sole determinant of how we age.
Right.
And it opened the door for other theories.
So if it wasn't just about bad genes, what else could explain aging?
Well, another big theory that emerged was the free radical theory of aging.
Okay, tell me about that one.
This theory pointed the finger at unstable molecules called free radicals.
Free radicals.
Yeah, these are molecules that are missing an electron.
Oh, like they're incomplete.
Exactly, and they're highly reactive.
Oh, I see.
They want to steal an electron from other molecules to stabilize themselves.
So they're just going around causing trouble.
And that damage can accumulate over time and contribute to aging.
So it's like rust on a car.
Right.
It just slowly eats away at it.
Exactly, and these free radicals are especially abundant in our mitochondria.
Mitochondria, right?
Yeah, powerhouses of our cells.
Right.
They're responsible for generating energy.
And so the theory is that as these mitochondria work, they produce these free radicals as a byproduct.
And over time, that damage builds up.
Okay, so I've got the mutation theory and the free radical theory, but it seems like neither of them fully explains aging.
Yeah, they both have limitations.
And this is where Sinclair really starts to lay out his own theory.
Right, he introduces this really important concept.
The epigenome. Tell me about that.
So imagine our genome, our DNA, as this complete musical score.
A musical score.
Yeah, like every note is there.
Exactly, and every cell in your body has the same skull.
The same DNA.
But a heart cell plays a very different tune than a brain cell.
That's right.
And that's where the epigenome comes in.
Okay. How does it work?
Think of the epigenome as the conductor and the musicians.
The ones interpreting the score.
Exactly. They determine which parts of the score are played how loudly, how softly.
I see.
It's the layer of control and instructions above the DNA itself.
So it's telling the cells what to do with that genetic information.
Right. And it's this epigenome that determines a cell's specific identity and function.
So the epigenome is basically controlling gene expression.
Yeah, it's deciding which genes are turned on and which genes are turned off.
And how does this all relate to aging?
Well, this helps explain why identical twins who have almost the same DNA can age so differently.
That's a great point, because they have the same score,
But one twin might experience more epigenetic changes.
leading to different outcomes.
Exactly. And studies have actually shown that genetics only accounts for a small percentage of how long we live.
How small are we talking?
Like somewhere between 10 and 25%.
Wow, so the vast majority of our lifespan is influenced by other factors.
By our environment and by these epigenetic factors.
So it's not just about the genes we inherit, but about how those genes are expressed and regulated.
And this leads us to Sinclair's central theory.
The big idea.
He calls it epigenetic noise.
Epigenetic noise.
Yeah, and it's this idea that over time, our epigenome starts to lose its precision.
So that conductor starts to make mistakes.
Yeah, the instructions get garbled.
So genes that should be on get turned off, and vice versa.
And this causes our cells to lose their identity and function less effectively.
It's like a slow decline in cellular organization.
She uses a great analogy here.
Oh yeah, what is it?
He compares it to a scratched DVD.
A scratched DVD.
Yeah, the information on the DVD, the genome is still there.
Right, but the player can't read it properly.
Exactly. And so that's what happens with our cells.
The underlying genetic information is there, but the instructions for accessing and using it become corrupted.
And that's what leads to aging.
Okay, but I had to ask, what's the optimistic side of this analogy?
The good news is that unlike a broken DVD where the information is gone forever,
Right.
a scratched DVD can be polished.
So you're saying that this epigenetic information might be recoverable?
Exactly. We might be able to restore those instructions.
And that's where the potential for reversing aging comes in.
Exactly.
So what are the key players in maintaining this epigenetic order?
The ones that get affected by this noise.
Sirtuins.
Sirtuins,
Yeah.
yeah, and these were a family of longevity genes.
Longevity genes.
Yeah, they were first discovered in yeast.
Yeast, like the stuff we use for bread and beer.
Yep, those tiny single-celled organisms.
I wouldn't have thought they could teach us much about human aging.
I know, it seems kind of strange.
Yeah.
But it turns out that a lot of the fundamental biological processes are conserved across species.
So what we learn from yeast, it can actually be relevant to humans.
Totally. And so these sirtuins, they're like the cell's internal quality control team.
Okay, what do they actually do?
They're enzymes, and their job is to regulate the epigenome.
How do they do that?
So they do this by removing certain chemical tags from proteins called histones.
Histones.
Yeah, these histones are like spools that our DNA winds around.
Oh, I see.
And by removing these tags, the sirtuins can control how tightly or loosely the DNA is packaged.
And that affects how accessible the genes are.
Exactly, and they're involved in really important processes like DNA repair and energy metabolism.
So they're keeping things running smoothly.
And here's the critical part. These sirtuins need a molecule called NAD to function.
NAD.
Nicotinamide adenine dinucleotide.
Okay, that's a mouthful.
I know.
But it's important.
Yeah, think of NAD as the fuel for the sirtuin quality control team.
So without enough NAD, the sirtuins can't do their job properly.
And the theory is that as we age, our NAD levels decline,
So our quality control team is running low on fuel.
which leads to more epigenetic noise and ultimately aging.
So that's the link between NAD and aging.
Exactly.
And you mentioned that sirtuins were first discovered in yeast.
Yeah, and what's interesting is that researchers found that increasing the activity of a specific sirtuin called Sir2 in yeast actually extended their lifespan, especially when the yeast had plenty of nutrients.
So by boosting sirtuin activity, they could make the yeast live longer.
Yeah.
Wow, that's fascinating.
I know, it's like a little hint that manipulating these pathways could influence longevity.
Even in complex organisms like humans.
Potentially.
So we've talked about yeast, but what about evidence for this theory in mammals?
Well, Sinclair talks about these experiments with mice called icy mice.
Icy mice.
Yeah, and it's a pretty compelling piece of evidence.
What do they do with these mice?
So scientists intentionally created breaks in the DNA of these mice.
They broke their DNA.
But these breaks weren't in the genes themselves.
Where were they then?
They were in the non-coding regions of the DNA.
The parts that don't directly code for proteins.
Right. And what was remarkable is that these mice started to show signs of premature aging.
Like what kind of sign?
They got gray hair, their spines started to bend, they even developed cloudy eyes.
And all of this happened at a relatively young age.
Yeah, like 16 months for a lab mouse.
Well, that's pretty young.
And remember, their genes hadn't been altered.
So what was causing this premature aging?
The theory is that these DNA breaks, they signaled to the Sirtuins that there was damage to repair.
So the Sirtuins were diverted from their normal duties.
Right. They were so busy fixing those breaks that they neglected their regular maintenance of the epigenome.
Leading to epigenetic noise and premature aging.
Exactly. And this experiment really highlights how important it is to maintain the integrity of our epigenetic information.
To keep those instructions clear and accurate.
It's not just about the genes themselves.
It's about how they're regulated.
And Sinclair also looks to nature for examples of organisms that seem to defy aging.
Oh, yeah. What are some of those examples?
Well, he talks about bristlecone pine trees.
Bristlecone pine trees.
Yeah, these trees can live for thousands of years.
Thousands of years.
And they don't show the usual signs of aging.
That's incredible.
I know. And then there's the Greenland shark.
The Greenland shark.
This shark can live for centuries.
Centuries.
And it doesn't even reach sexual maturity until around 150 years old.
Wow, that's just mind-boggling.
I know these examples, they suggest that the rate of aging isn't fixed.
That it can be slowed down significantly or maybe even reversed.
And that's a really exciting idea.
It is. It opens up so many possibilities.
So given all of this, what can we do to influence our own longevity?
That's the big question.
Sinclair talks about activating what he calls our ancient survival circuit.
Our ancient survival circuit.
Yeah, these are genes and pathways that have evolved over billions of years to help us survive tough times.
Like when food is scarce, where the environment is harsh.
Exactly. And these genes like FOXO3 have been linked to longevity.
So they're kind of like our built-in longevity genes.
Right. And one way to activate these genes is through something called hormetic stressors.
Hormetic stressors.
Yeah, these are beneficial forms of stress.
Beneficial stress.
I know it sounds counterintuitive.
Yeah, how can stress be good for you?
Well, think of it as challenging your body in a controlled way.
Like a workout.
Exactly. Exercise is a prime example.
So putting our bodies under physical stress can actually make us stronger and more resilient.
Yeah, especially vigorous activity that gets you breathing hard.
That creates a temporary lack of oxygen.
A hypoxic response.
And that triggers these survival mechanisms.
And what other hormetic stressors does he talk about?
Well, he mentions calorie restriction.
Calorie restriction.
Yeah. Studies have shown that reducing calorie intake without malnutrition can extend lifespan in various animals.
Really? Even if you start in middle age?
That's what the research suggests.
Wow, that's pretty amazing.
And he also discusses intermittent fasting.
Intermittent fasting.
Yeah, which kind of mimics some of the effects of calorie restriction.
So you're basically cycling between periods of eating and not eating.
Exactly. And he also mentions exposure to cold temperatures.
Like taking cold showers.
Yeah. Or ice baths.
I don't know if I could do that.
It's definitely not for everyone.
But the idea is that these cold temperatures, they also activate those survival pathways.
They challenge the body and force it to adapt.
So it's all about finding those healthy challenges.
Right, those things that push us just outside of our comfort zone.
But not too far.
Exactly.
And what about things like supplements or medications?
Well, he does explore some pharmaceutical interventions.
Like what?
He talks about rapamycin.
Rapamycin.
Yeah, it's an antifungal compound that was originally discovered in the soil of Easter Island.
Easter Island?
I know, it's a pretty remarkable story.
How does it work?
Well, it inhibits a pathway called TOR.
Q.
Which is involved in cell growth and metabolism.
And inhibiting TR has been shown to extend lifespan in various organisms.
Yeah, it's a pretty promising area of research.
And he also mentions metformin.
Metformin.
Yeah, which is a common drug used to treat type 2 diabetes.
Right, and studies suggest that it might also have anti-aging properties.
Really?
Yeah, it seems to affect the mitochondria.
The powerhouses of our cells.
Exactly. So it's kind of like giving them a tune-up.
Okay. And then there's all this buzz around NAD boosters.
NAD boosters.
Yeah, since NAD levels decline with age, and it's so crucial for sirtuin function.
The idea is to replenish those NAD levels.
Exactly. And Sinclair talks about molecules like NMN,
NMN.
nicotinamide mononucleotide.
He's actually experimented with NMN himself.
He has, and he talks about his own experiences and how his father seemed to rejuvenate after taking NMN.
It's a really interesting anecdote.
But he's also careful to point out that the research on NMN is still very preliminary.
Yeah, it's important to be cautious about drawing conclusions from anecdotal evidence.
Totally. And he also mentions some very early unpublished findings in animals that suggest potential benefits for restoring things like menstruation and fertility.
Yeah, that's pretty remarkable.
But again, it's very early stage research.
Yeah, we need more studies to confirm these findings.
And to determine if it's safe and effective in humans.
Absolutely.
So we've talked about boosting NAD, but what other pharmaceutical strategies are being explored?
Well, another promising area is senolytics.
Senolytics.
Yeah, these are drugs that target and eliminate senescent cells.
Senescent cells.
These are basically zombie cells.
Zombie cells.
Yeah, they stop dividing, but they're still hanging around, and they can release harmful molecules.
Like inflammatory molecules.
Exactly. And that can contribute to aging and age-related diseases.
So by getting rid of these zombie cells, we might be able to slow down aging.
That's the idea. And early human trials with senolytics are already underway.
For things like osteoarthritis and glaucoma.
Exactly. So it's a very active area of research.
Okay, so we've covered a lot of ground from lifestyle changes to pharmaceuticals, but perhaps the most revolutionary idea in the book is cellular reprogramming.
Cellular reprogramming.
Yeah, and this is like a potential game changer.
How so?
Well, it's inspired by the work of John Gurdon and Shinya Yamanaka.
Who are they?
They're scientists who showed that mature, specialized cells can be reprogrammed back to a more youthful stem cell-like state.
Wow, so you're saying we can essentially turn back the clock on our cells?
Yeah, and this technology could allow us to reset the epigenetic landscape of our cells.
To erase those scratches on the DVD.
Exactly, and imagine being able to regenerate damaged tissues or even entire organs.
That would be incredible.
I know it's like science fiction, but it might become reality.
So this deep dive into lifespan has been quite a journey.
It has. We've gone from the basics of biology to these cutting-edge interventions.
What stands out to you as the most significant takeaway from Sinclair's work?
For me, it's this shift in how we view aging.
How so?
It's not just this inevitable decline.
Right.
It's a loss of information, a breakdown in how our genes are regulated.
And that opens up the possibility of reversing aging.
Exactly, and he gives us all these potential strategies for doing that.
From lifestyle changes to pharmaceuticals to this mind-blowing idea of cellular reprogramming.
What's the biggest aha moment for you?
The idea that we might be able to reverse aging at a fundamental level through cellular reprogramming is just revolutionary.
It really challenges our assumptions about what's possible.
It does. It makes you think about the future in a whole new way.
And that's what Sinclair's work does. It encourages us to question the idea that aging is just something we have to accept.
To see it as a process we could potentially influence.
And he paints this compelling vision of a future where living longer and healthier lives might become the norm.
That's a future I'd like to see.
Me too. And so this brings us to a final thought for our listeners.
What's that?
With this knowledge, what aspects of your own health and lifestyle might you reconsider?
That's a great question.
Yeah, it's really something to think about.
Thank you for joining us on this deep dive into lifespan.
Yes, we hope this has been a valuable shortcut to understanding this complex and fascinating topic.
And we hope it sparked your curiosity to explore this field further.
So please subscribe to the Nonfiction Insights channel for more deep dives into thought-provoking books.
We'll see you next time.
Bye.